Greetings and welcome back to my new series of lesson on the 5th chapter titled Principles of Inheritance and Variation. At the outset, let me draw your attention to the learning goals of this session. 1. Define the terms genetics and heredity. 2. Name the father of modern genetics. 3. Identify and explain the widely accepted theory of inheritance during Mendelian time. 4. List the 7 contrasting traits selected by Mendel in pea plants. 5. Analyze the advantages of choosing pea plants for genetic experiments. And 6. Determine the 2 types of genetic crosses conducted by Mendel. Well, it was known to mankind since ancient time that traits got passed on from parents to the offsprings, and the offsprings that are obtained through the process of sexual reproduction always showed variation. Not only that, many farmers and breeders have manipulated plants and animals for thousands of years in order to create new improved varieties of crops and animals. However, the science behind the mechanism of inheritance was unknown for a long period of time. It was not until the mid-19th century that a breakthrough was achieved in the understanding of inheritance of traits. The term genetics actually was coined in the year 1906 by an English biologist named William Bateson, and this term was to designate to the new science of heredity. So what is genetics? Genetics refers to a branch of biology that deals with the study of heredity and variation in living organisms. Let's also be clear with the other 2 terms here with reference to genetics, that is heredity and variation. Heredity or inheritance is a term that refers to the passing on of traits from parents to the progeny. Or in another way, we can say that this is basically referring to the passing on of the traits from one generation to the next generation. And variation refers to the degree of differences that exist between individuals. Or you can also say that this is the difference that we can find between progeny and their parents. So what is the history of genetics? Before the idea or concept of DNA, chromosome or gene actually existed, a monk in the garden had pioneered to understand with the basic pattern of inheritance. And he was Gregor Johann Mendel. And his contribution has become the foundation of modern genetics. For this reason, Mendel is rightly known as the father of modern genetics. Now, to give you a brief account on Mendel, Gregor Mendel was an Austrian monk born in the year 1822. He belonged to a farming family, and his love for nature most probably stems from his early rural experiences. Now, despite the financial strains and his health issues, Mendel was able to graduate from high school. And at the age of 21, Mendel decided to join to the local Augustinian monastery against his father's wish with the hope to pursue with his education. So while at the monastery, Mendel received funding to do his further study in the University of Vienna. So at the University of Vienna, he studied physics, mathematics and plant physiology in particular. And after completing with his 2 years program, Mendel returned back to the monastery and perceived with his passion in teaching as well as in many of the sciences. Now, Mendel was very curious about how traits are inherited from one generation to the next generation. And he then set out to understand the basic principles of inheritance by conducting various experiments in the monastic garden. So from 1856 to 1863, Mendel ultimately decided to work on the garden pea plants. And his experiments or his research on the garden pea plants were primarily based on hybridization experiments. So through his hybridization experiments, Mendel was able to understand or to study the various aspects of inheritance, and finally he was able to be proposing with some of the basic principles of inheritance. Well, during Mendel's time, one of the theories which became very popular and widely accepted was the theory called as blending inheritance theory. Now, according to blending inheritance, it proposes that the traits of the parents are going to be getting mixed together, and then they are going to be transmitted to their offsprings. Now, say for example, if we consider flower color as the traits in the parent. So, if we have two of the parents who have red flower and white flower. So, let's say one of the parents, like the male parent, for example, has red flower, and the female parent, for example, let's say has white flower. Now, according to blending inheritance, what is to be transmitted to the offspring is, it has to be a blend of these two colors. That is to say, the flower color in the offspring should be an intermediate of the flower color of the two parents. So, in this case, because we have a red and a white flowered parent, so these two parents are to be blending and giving rise to a intermediate flower color, which is a pink flower. So that is how we can understand that the traits are to be inherited by mixing together and then getting transmitted to the progeny. And this theory of inheritance is referred to as blending inheritance theory. Now, in order to study with the principles of inheritance, Mendel selected 7 pairs of contrasting traits in pea plants. And these 7 traits were stem height, seed color, seed shape, pod color, pod shape, flower color, and flower position. Now, when we say that these traits had contrasting nature, what we mean here is that each of the traits have 2 contrasting or opposing forms of characteristics. For example, stem height can either be tall or dwarf. Likewise, seed color can either be yellow or green. Seed shape can either be round or wrinkled. Pod color can either be green or yellow. Pod shape can either be inflated or constricted. Flower color can either be purple or white. Flower position can either be axial or terminal. So I strongly recommend that you learn these 7 pairs of contrasting traits because they are going to be forming the framework of Mendelian genetics. Now, isn't this very cool? I feel Mendel had been very smart to have chosen the traits that showed very distinct observable characteristics. And with this kind of the contrasting forms of the characteristics, it becomes highly easy with no room for any sorts of ambiguity or vagueness while making observations of the traits in the progeny. Next, let's figure out why Mendel chose pea plants for his hybridization experiments. Pea plants are a good choice for Mendel because, firstly, they are easy to raise and fast growing. This would enable Mendel to study many generations in a short period of time. So, the first benefit here is it enables to study many generations in a short span of time. Secondly, they have many variations with contrasting traits. Now, as I said earlier, such type of distinctive observable characteristics would allow Mendel to make easy and accurate observations. So, let's see, it can make easy and accurate observations. Thirdly, they can be self-pollinated as well as cross-pollinated. Now, this is of great importance to Mendel because, especially cross-pollination. Because Mendel can be performing controlled pollination and thereby he can be making crosses between plants of his choices. So, this can perform controlled pollination. As well as and make crosses of his choice, which is very important for any kind of hybridization experiment. And then they also produce huge amounts of seeds, thereby raising with or facilitating with a larger sample size, right? And whenever there's a larger sample size, the credibility to the result always increases. So you can say it facilitates larger sample size because of huge number of seeds which are available, and thereby it adds greater credibility to his results. So all in all, these characters are responsible for the successful completion of Mendel's hybridization experiment. Well, how did Mendel go about with his hybridization experiments in pea plants? First of all, Mendel established true breeding lines or pure lines in all the 7 pairs of contrasting traits. Now, what do we mean by true breeding line? A true breeding line refers to a variety or a plant in this case that would always be expressing a stable character from generation to generation. Now, to get a better idea of this, let's take an example. For example, let's say that we have a true breeding tall pea plant. Okay? So we have a tall pea plant, and this tall pea plant, right? The character here is the tall-heighted nature of the pea plant. So this tall pea plant, being a true breeding line, will be producing offsprings that are also going to be expressing the tall height character. Okay, so it's going to be producing all tall pea offsprings. Alright, so a tall pea plant having all of its children or offsprings which are also tall in nature. And if we make a kind of selfing between the offsprings or between the children, they are also to be giving rise to all tall pea plants. So what we see here is the character that was present in the parent plant, which was the tall-heighted nature of the parent plant, is always expressed in all of the subsequent generation. Okay, so from generation to generation, we find that the character remains stable or the character remains intact. The character remains identical to that of the character that was there in the parent plant. So such kind of a phenomenon or such kind of a variety where we say that the variety is always going to be producing its own type. The variety is always going to be producing offsprings that are identical to itself. Such kind of varieties are referred to as true breeding line. Okay, and Mendel was able to be establishing true breeding lines for all of the 7 pairs of contrasting traits. You might wonder how Mendel established these 7 pairs of true breeding lines. Now this was primarily possible because pea plants are naturally adapted for self-pollination. Now if you look into the structure of the pea flower here, what you see is that the petals are arranged in such a way that they almost enclose the male and female reproductive structures. And because of which, this would always ensure higher rates of self-pollination and thereby maintaining with the purity of character. And with this kind of phenomenon, what Mendel did was Mendel grew such plants obtained through continuous self-pollination for several generations until he was able to be arriving at the true breeding variety. Which means to say now the plants are always producing offsprings identical to itself. And in fact, it took about 2 years for Mendel to achieve all of these 7 pairs of contrasting traits. After establishing with the true breeding lines, Mendel then went ahead with his actual genetic crosses in order to understand with the principles of inheritance. And Mendel's genetic crosses can be basically grouped into 2 main types of genetic crosses or hybridization. The first one is known as monohybrid cross and the second one is known as dihybrid cross. Now what we understand by monohybrid cross? This is a cross which is between 2 plants that differ just in one trait. Whereas the second type of cross, which are known as dihybrid cross, in these cross, the 2 plants, parent plants are to be differing in 2 traits at a time. Okay. Now to understand this better, let's again take the typical examples of monohybrid and dihybrid crosses. So for a monohybrid cross, let me say I'm going to be taking a tall pea plant as one of the parents. Okay, so you can be considering this as a male or a female parent. So I'm going to be taking the tall plant as, let us say, the male parent, and this male parent is to be crossed with another parent differing in this character. Okay, so just because the male parent is a tall pea plant, now the female parent to be selected or to be used here to be having the opposing form of a tall character, which is dwarf. So this is crossed with a dwarf pea plant. So such kind of a cross is known as monohybrid cross. The reason is because the 2 parents that we are having here are basically differing only in one trait. And in this example, the trait that they are differing in is stem height. One is tall-heighted, the other is dwarf. Right? For the dihybrid crosses, because here we have to consider 2 traits at a time. Okay, so now these are the parent plants are to be differing in 2 traits. So let me take a typical Mendelian example as well. So let's say that we are going to be crossing a round and yellow seeded plant, which you can be considering either as a male or a female parent. So for in this case, let me say this is a female parent. Okay, so the female parent has, we are considering 2 of the characters at a time. So this female parent has round shape of the seed as well as yellow colored seed. Alright, now this is to be crossed with another male parent, and that male parent should be differing in these 2 characters to the female. Alright, so as we know, the seed shape can be either round or wrinkled. So in the male parent, we are going to be having wrinkled seed, and instead of the yellow seed color, we are going to be having its opposing, the contrasting form, which is green seed color. So this is how we can be taking the example for a dihybrid cross, where these 2 parent plants are to be differing in 2 traits simultaneously. And in this example here, what we have taken is we have selected the parent plants differing in seed shape and seed color. So the seed shape here is round and the other parent has wrinkled seed shape. The seed color here in one of the parents, that is in the female, is yellow, and the seed color of the male parent is green. And because we are having two of the parents differing in two traits, such kind of crosses are known as dihybrid crosses. Now, Mendel could be achieving these kinds of monohybrid and dihybrid crosses through controlled pollination, or we can say through artificial hybridization. All right. In this regard, what we also want to be realizing is, in order to make such kind of hybridization crosses, or you can say in order for Mendel to be making such kind of selective crosses, he had to do many controlled pollination. The reason is because pea plants have bisexual flowers, and as we discussed earlier, that these flowers are adapted naturally to have more of self-pollination. Now, if Mendel have to be conducting crosses of his or making crosses of his choices, just let's take an example here. If Mendel have to cross a male tall plant with that of a female dwarf plant, now he have to make sure that the stigma of these dwarf pea plant are not pollinated or by its own pollen grains. Right? And to prevent such self-pollination, there are basically many different kinds of outbreeding devices. We have learned this in the previous second chapter. So, two of the major techniques here are emasculation and bagging. So, what Mendel had to do was, because the dwarf female plant was to be pollinated by preferred tall pea plant, so he emasculated the anthers of these dwarf pea plants prior to their maturation, right? And then these emasculated flowers were bagged for a certain period of time. Now, once the stigma got mature, then he had to collect the pollen grains, and he did it by himself, by hand, right? So he collected the pollen grains of the male tall plant, and then painstakingly dusted that over to the stigma of the dwarf pea plant, which was his female parent here, right? And in this way, he was able to perform controlled pollination, right? And this was not just for one or two cross. He conducted such kind of controlled pollination for about 30,000 crosses. Can you imagine? He conducted such kind of controlled pollination by himself for about 30,000 crosses in the pea plants, right? And in that way, with this kind of a very laborious, a very intensive, you can say, work by Mendel, Mendel was able to be finally coming up with, or finally making some kind of inferences from all the different kinds of patterns that he saw with such numbers of crosses. Here is an overview of Mendel's work on pea plant. To start with, Mendel established true breeding lines for all the 7 pairs of contrasting traits. And using these true breeding lines, Mendel then went ahead with his hybridization experiments or genetic crosses. Now, all of these genetic crosses were possible because of controlled pollination, or you can say it was done by emasculation. And Mendel broadly had two main types of genetic crosses, monohybrid cross and dihybrid cross. Now, the way Mendel made his observations of all of these almost 30,000 crosses was chiefly based on the physical characteristics or the phenotypes, the external features, which we technically called as phenotype of the progeny. And in addition to the observation of phenotype of the progeny, Mendel also counted the numbers, the exact numbers of the plants possessing a particular trait. So in this way, what you can see here is that Mendel had been very systematic and very methodical in terms of his execution of the hybridization experiment. Apart from that, Mendel also used statistical method in order to analyze and predict his experimental result, which was actually a breakthrough or a kind of unheard of approach to dealing with a kind of biological phenomenon. So in this way, Mendel was able to complete his work successfully, and in the year 1865, Mendel presented his work. Mendel presented his work to the local Natural History Society in the year 1865, and in year 1866, Mendel published his observation on the principles of inheritance in a paper titled Experiments in Plant Hybridization. However, Mendel's work went largely unnoticed or unrecognized during his time. It was not until 1900 when three of the European biologists, namely Hugo de Vries, Carl Correns and Schermack, rediscovered Mendel's work. All the three of them worked independently on different plant hybrids and helped to expand Mendel's work of inheritance to the scientific world. Unfortunately, Mendel was not alive to receive the recognition as he died in the year 1884. To this day, Mendel's principle forms the basis to explain the various phenomena of inheritance. So with this, I come to the end of today's lesson, and I'll see you again with my next video on monohybrid and dihybrid cross.